Can you hear me? Yes, yes. Morning, ladies and gentlemen. I will change a little bit the focus compared to the first speaker. So my presentation will be more on an academic topic. So I will try to convince you of the interest of peptides that interact with multiple pharmacological targets. And I will describe our efforts to develop such multi-target peptides for the treatment of neuropathic pain. For many years, the pharma industry has focused on the development of magic bullet drugs, following the idea, one disease, one target, one molecule. So the molecules, the drug molecules were designed to have high affinity for this target and also high selectivity, because any off-target interaction was associated with side effects. Now, this approach has been very successful for a number of diseases. However, it is also correct that many complex diseases remain inadequately treated. And therefore, in recent years, the concept of polypharmacology is attracting a large interest. The basis for polypharmacology is the observation that many complex diseases like cancer, neurodegenerative diseases are multifactorial. And the concept of polypharmacology can be very easily illustrated in this simple drawing. So suppose this is a town center which is very congested because too many cars are arriving through the main road. Then you can correct this situation by blocking the main road, and for a while the situation will be better. It will be better until the drivers find alternative ways to get into the city center this way. And then you have the same situation again. So what actually you have to do is to block multiple pathways. And that is polypharmacology. So you can do that by administering multiple drugs, multiple of these selective drugs that block each of these pathways. That means you are giving a drug cocktail. So in a drug cocktail, the patient has to take several tablets during the day with different components. Now, these different components may have different pharmacokinetic properties, like a different rate of metabolism, different excretion. So that gives a complex PK situation. It also results in difficult patient compliance. And because you give several compounds, you have the risk of drug-drug interactions. An alternative is, of course, that you formulate multiple components into one tablet, but it doesn't change to the pharmacokinetic problems or to the drug-drug interaction, evidently. It's just easier for the patient. The easiest solution is actually that you can give one single compound called a designed multiple ligand, and this compound should interact with the different drug targets simultaneously. Because it's only one compound, there is only a single pharmacokinetics. There is no risk of drug-drug interaction. In contrast, you can expect synergism, less toxicity, increased efficacy. Because you have only one compound, the challenge is actually to build into that compound the right relative potency for the different drug targets. And that is very, very challenging. So how do you design such a multiple target ligand? The simplest way is that when you have two compounds with two pharmacophores, you link them through a spacer. Then you have linked pharmacophores. If the spacer gets shorter or inexistent, you may have fused pharmacophores. And if the two pharmacophores have some common feature, you can overlap these common features, and then you get a merged pharmacophore. Of course, these are the most interesting because they have the smallest molecular weight. They are most drug-like. When you do this, you have to consider the binding mode of these multiple ligands. When you have two receptors, like here I show two G protein coupled receptors, and you want two compounds to interact simultaneously with the two receptors, you have to consider that there is a minimum distance between the two active sites. So when you link the two, and you want them to bind simultaneously with the two receptors, you need a quite long linker, like 30 to 60 atoms long, which gives you quite complex molecules. If the linker is shorter, like here, only one of the two components can bind to its receptor. Then it has to dissociate in order for the second component to be able to bind to the second receptor. So this is a bivalent mode. That is actually a bifunctional mode. It's not the same. And evidently, when you have a fused, or a merged pharmacophore compound, it can only bind in the bifunctional mode. So these merged multi-target drugs are the most interesting. A recent review in beginning of this year stated that actually the design of such compound is still in its infancy. And especially for small molecules, excuse me, it is quite challenging. However, peptides are extremely suited for this kind of exercise. Linking or fusing peptides is quite easy. Peptides usually have quite extended pharmacophores consisting of amino acid side chain. And so very often you can find some common feature between the different pharmacophores and overlap. So when they allow when judiciously chosen, a good overlap. And still, even in peptides, this adjustment of the relative potency, what is the the most potent component compared to the other, is still challenging, but as I will illustrate, it is feasible. I will illustrate this concept with the development of new analgesics for neuropathic pain. Pain is evidently an important problem which has an impact on people and on society. It is estimated that about 100 million adults in the US suffer from persistent pain. This translates to 1.5 billion people worldwide. The cost is quite substantial, and actually the cost of pain exceeds the economic costs of the 6 most costly major diagnoses, like cardiovascular diseases and neoplasm. Severe pains, especially severe acute and chronic pain, is mostly treated now by giving opioids like morphine. However, morphine is not effective for neuropathic pain. Neuropathic pain is actually pain resulting from nerve injury by trauma, surgery, disease, and so on. In chronic pain, one has to administer opioids regularly, and this leads to tolerance, which means that actually the patient gets less sensitive to the analgesic effect of morphine. Then you have to increase the dose. But when you increase the dose, you also increase the side effects, like, for instance, constipation, respiratory depression, addiction, and so on. Importantly, it has been demonstrated that persistent pain upregulates a network of anti-opioids. These are compounds that actually counteract the effect of opioids. in a network as I described. So opioids can be divided in non-peptidic like morphine, which is a natural compound, I'm sure you know, synthetic like fentanyl, and then you have peptides, endogenous, which we produce ourselves, like the endomorphins, or exogenous, like deltorphin C and the morphine. So these are isolated, actually, they are very potent opioids. They are isolated from the skin of a South American frog, this one here. All of these opioids interact with the so-called opioid receptors. You have three major types, the mu, the delta, and the kappa opioid receptors. And it is very important to realize that these opioid receptors are located in the brain. So any analgesic acting at opioid receptors should be able to get into the brain and thereby cross the blood-brain barrier. From previous structure activity studies, it has been postulated that the best or the desired profile for a good opioid analgesic would be a balanced mu-delta agonist, so acting at the mu and the delta opioid receptor with about equal potency as agonists. Because the administration of opioids upregulates a number of anti-opioid pathways which diminish the effect of morphine, one of the approaches was to develop compounds which are hybrids of opioids and anti-opioid components. For instance, one of these major anti-opioids is substance P, and you have also CCK. So substance P counteracts the analgesic effect of opioids. Substance P interacts with the NK1 receptor, which is also upregulated in persistent pain. And therefore, when you have an NK1 antagonist, you block this receptor, that should potentiate the effect of the opioids. And therefore, people like Lipkowski have developed hybrid compounds where you have an opioid agonist pharmacophore, this part, and an NK1 antagonist pharmacophore, that part. The same was done with Largent Miles, opioid agonist pharmacophore, NK1 antagonist. And the same was done for CCK. It also attenuates morphine-induced analgesia. So you have here an opioid agonist with a CCK antagonist pharmacophore. So these hybrid compounds acting at two different pathways were shown to be more efficient for the treatment of pain than just the opioid paths by themselves. Another approach is to use an opioid pathway together with a non-opioid pathway for analgesia. For instance, neurotensin is a peptide that induces analgesia, but not mediated by the opioid receptor. So when you combine an opioid pharmacophore with a neurotensin pharmacophore, you act at two different pathways, which gives you a more efficient analgesic. Now, we have used a different approach. We wanted to make hybrid compounds with actually trifunctional hybrid compounds, which were mu opioid agonist, delta opioid agonist, NK1 antagonist hybrids, and we wanted to be them as compact as possible. We started from the natural compound dermorphine, isolated from the South American frog, and we used the minimum sequence, which is the N-terminal tetrapeptide here. We looked at known NK1 antagonist structures, and we tried to search for common pharmacophore between the NK1 antagonist and the peptide. And the basis for this search for this common pharmacophore structure was actually the use of conformationally constrained aromatic amino acids. So here you have the structure of phenylalanine. There is a lot of rotational freedom about this single carbon-carbon bond, so the side chain is very flexible. But we constrain the peptide by making a bridge here between, we bridge the aromatic ring, and we link it to the nitrogen of the amide backbone here. You can do that for phenylalanine, tryptophan, something that looks like histidine. So what is the basis for this concept? Well, this is the flexible peptide. The flexible peptide can adjust its main chain conformation to fit into different receptors. So it can have this form, that form, and fits into different receptors. It can also adapt its side chain conformation to fit into the pockets, like here you have one orientation, there you have the other one. If you fix this side chain orientation by linking it to the main chain, like as is shown here, you can see that it will not fit into this receptor anymore. So when these receptors give you the side effect, you get rid of the side effect and you get still the affinity for the target receptors that you want. Moreover, we hope that by using this constrained scaffold, we would be able to generate sufficient binding affinity to get high affinity compounds. Excuse me. So we did this approach starting from this morphine tetrapeptide, and I'm showing here the affinity for the opioid receptors. These are IC50 values. If you are not familiar with these values, the lower the number, the better. So you can see here that this tetrapeptide has a much better affinity for the mu opioid receptor than for the delta opioid receptor. When we incorporate these side chains into a ring, like here, you see we maintain the mu affinity, we gain a lot in delta opioid receptor affinity. This is a balanced mu opioid activity. That is the profile that we are looking for. But still, the potency is not quite high. It's 20 nanomolar. So we cut open the ring here again, but we left the methyl substituents. So we got this compound, BVD3, which is really a quite potent, balanced mu delta opioid agonist. These compounds were tested in vivo for their analgesic effect, and for that the mouse tail flick assay was used. In this assay, the tail of the mouse is immersed into a hot water bath, 55 degrees, and normally the mouse just flicks its tail out of the water because of the hot water. When you give an analgesic, the mouse leaves its tail much longer, and this time is measured. The maximum time is 10 seconds in order not to harm the tissue of the animal. So 10 seconds is the maximum possible effect. And the other times are mentioned as a percentage of this maximum positive effect. Okay, so the red bars here are the bars for morphine given at 2 milligrams per kilogram. You see after 5 minutes the influence of morphine. Then it has almost maximum effect after 15 minutes, goes down after 16 minutes. This compound is SB, that is the curse for this compound, at a dose of 2 milligrams per kilogram here, after 2 hours is equipotent to morphine, but its onset is slower. In contrast, that compound here, when you look after 2 hours, here the morphine is the purple bar. After 2 hours, this dose, which is 0.5 milligram per kilogram, is even more potent than morphine. So this is a quite potent compound. It is also important to mention that the compounds are given intravenously. Um, that means that these compounds are able to cross the blood-brain barrier because they have to exert their analgesic effect by interacting with the opioid receptors in the brain. So we have peptides that are actually able to cross the blood-brain barrier. We did the same exercise for another peptide called DMT-DALDA, which is this one. It's a very potent mu opioid receptor. You see the sub-nanomolar affinity. We constrain the aromatic ring here. We gain again in delta affinity, but the compound became a partial agonist, which is something we didn't want. We want a full agonist. Fortunately, when we omitted the lysine side chain, got the glycine, again we get a quite potent balanced mu delta agonist. Was tested in vivo, the same mouse tail flick assay, and it was compared to the previous one. So this is AN81, the blue bars. This is morphine, and that was the BVD3, the compound I showed in the previous slide. So the blue bars are a little bit higher than the purple ones. So AN81 is slightly more potent than the BVD3. So we use that compound in our further studies. We also tested whether this compound induced tolerance. So that means that when you administer the compounds for several days, then when the mice develop tolerance, the analgesic effect will go down. So here you see the effect of this AN81 after 5 days, and the effect is almost gone. So it means it's a potent analgesic, but it induces tolerance. So now the NK1 part, because we are looking for hybrids between opioids and NK1. As I said, we looked at the literature for known NK1 antagonists like this. We constrained them by making the cycle here and here. Unfortunately, all these compounds were inactive. We then turned our attention to another class of known NK1 antagonists, which have a benzodiazepine structure. And it was reported that actually the two aromatic rings here were the important pharmacophore elements. So we thought we could display these on the constrained scaffold derived from phenylalanine that we developed. But unfortunately, this compound is also inactive. Then we left out the phenyl, and we got a compound that has reasonable affinity for the NK1 receptor and was a quite good NK1 antagonist. Interestingly, when you change the phenyl for the indole, it's inactive. But anyway, we had now also a good NK1 antagonist. So at the left part is the opioid part, starting from the dermorphine tetrapeptide. We developed AN81, potent and balanced mu delta opioid. At the right side, we started from the benzodiazepine. Lead developed this NK1 antagonist, and then we overlap the common features and get this compact opioid agonist, NK1 antagonist. The hybrid compound maintained quite good opioid potency. It was also quite high affinity for the NK1 receptor. It is a bit less potent NK1 antagonist. So it was again tested in vivo. At day 1, it is a quite potent analgesic. In this mouse tail flick given intravenously, so the compound crosses the blood-brain barrier. However, at day 5, again, the analgesic effect was gone, illustrating actually that the NK1 antagonist component in this model, which is an acute pain model, does not have a beneficial influence on the analgesic effect. The reason could be that maybe the relative potency of opioid versus NK1 was not the right one that we needed. It might also be that we had to change from the mouse tail flick test, which is an acute pain test, to a neuropathic pain model. So to change the opioid NK1 balance, we did a number of structure activity studies. We modified quite a number of residues in this hybrid. And starting from the lead here, this was the SB compound, we obtained this one, where actually the glycine is changed to a beta-ALA, and the two trifluoromethyl groups have been removed. It is a weaker NK1 antagonist, but a more potent opioid. In parallel, we developed another class of compounds, this one where we had a weaker opioid, but more potent NK1 component. We don't have full data on this series of compounds, so I will discuss only this class of compounds. These compounds were tested now in a rat model for neuropathic pain. That is the chronic constriction injury model. In this model, the sciatic nerve of the animal is ligated here by 4 ligations. And 7 days after, these rats develop an increased sensitivity to normally noxious stimuli, which is called hyperalgesia, and also to normal non-noxious stimuli, which is called allodynia. And 7 days after the surgery, the rats are tested for this hyperalgesia or allodynia. The test used for hyperalgesia is a cold plate test, so the rat is put on a plate, 5 degrees Celsius, and the time is measured that it lifts the hind paw. The allodynia is measured in the so-called von Frey assay, where a filament, the von Frey filament here, is pushed onto the hind paw of the rats, and again, the reaction is timed. So here you see the effect of the hybrids. This is one with the beta-alanine, the Kg compound, compared with this one with the glycine, the SB compound. These are the hybrids, and this is the parent opioid. So in naive rats, so normal rats, using the tail flick assay, which is an acute pain model, you can see here the bars, which give you the maximal positive effects. This is the hybrid. This is the opioid part. Now, it's maybe difficult to see the doses, but this is a 0.1 milligram per kilogram for the opioid part. This is the same 0.1 for the hybrid. So meaning that the opioid part by itself is much more potent than the hybrid. And the same for this one. This is the opioid part, 0.1 milligram per kilogram compared to the hybrid. So in an acute pain model, it is clear that the opioid part is more potent than the hybrid. Now what about the neuropathic pain model? Well, here you can see the reverse. So you see now that here the hybrid, this one, is much more potent than the opioid. And the same for that one. The hybrid here is much more potent than this one. This is also reflected in the ED50. The ED50 is 0.30 nanomole per kilogram for this compound, 1.78 for that one. So they are more potent in this neuropathic pain and much more potent than morphine. which has an EC50 of 821 nanomole, and the same effect is observed in allodynia. So we were very pleased now that we had good compounds that were potent analgesics in a neuropathic pain model, and the hybrids were more potent than the opioids. So what about tolerance? Well, unfortunately, here you can see the time course of the analgesic effect as a function of the day of administration, and you can see at the first day you have a quite potent compound, but in the following days the effect goes down, just like for morphine. That is in the von Frey assay, that's in the cold plate assay. So unfortunately now we have good compounds which are good analgesics, but we did not solve the problem of the development of tolerance. So in conclusion, we have developed peptide analogs which are potent analgesics in a neuropathic pain model. Very important also for peptides is that these analogs are able to cross the blood-brain barrier, because when you give them intravenously, they have a central mediated effect. The conformational constraint amino acid analogs that we used are actually the key to the success of the design of these hybrid compounds. And the relative potency in the hybrids could be modulated, which is also an important aspect. And it was actually shown that the compound that is the more potent one, the Kg compound compared to the Sb compound, is more potent despite that it is a less potent NK1 antagonist, showing the importance of the relative potency of the two components. Unfortunately, the development of tolerance was not avoided. This is in contrast what is reported in the literature, especially by the Ruby group, but we were not able to confirm that observation. And again, I hope I have convinced you that peptides are a very suitable molecule for this kind of exercise to develop such multi-target compounds. I thank all my collaborators, especially Stephen Ballard, who is now the new head of the department, the students who did the work, and evidently there was a lot of collaborations for the determination of the pharmacological effects. I thank, of course, people who gave us the money to do the work. A special thanks to Senn Chemicals, who gave me the opportunity to present this work here, and I thank you for your attention. Thank you.